Hello everyone, myself Ms. Divya Sharma. I'm a second year B.Pharm student currently pursuing my degree in Viva Institute of Pharmacy. So today I'm here to shed light on the groundbreaking technology known as touch DNA, or revolutionizing evidentiary in DNA forensics. As we all know, every individual has their own unique DNA. It's a genetic fingerprint and it is an important aspect in crime investigation. The importance of touch DNA lies in its potential of linking an individual from the crime scene or any such evidence which is found there. In past, DNA analysis required a significant amount of biological material like blood, semen, tissue, or salivary samples. But now, touch DNA has brought a revolution in the system. The science behind touch DNA is really fascinating. Touch DNA particularly refers to the availability or presence of DNA cells on the object or surface which is transferred by direct contact or touch of person's skin cells from epithelial tissue. Therefore, it is also known as contact DNA or transfer DNA. The availability of this transfer DNA is possible on clothings, wearings, then on door handles, on weapons, tools, on personal belongings. By collecting and analyzing this trace DNA, investigators can actually bring a legal evidence between suspect and the crime. This novel technique of analysis includes extraction, collection, extraction, amplification, and further analysis of DNA. The extraction or the collection can be done by various methods like swabbing, scraping, by tape lifting, and it can further be extracted by automated DNA devices or by organic extraction. Then it is further analyzed for next generation sequencing. As we all know, DNA can naturally replicate itself. Even the great economist Kenneth Boulding has quoted that DNA was the first three-dimensional Xerox machine. So synthetically it is replicated by polymerized chain reactions and many such techniques, and it helps in generation of the complete DNA sequence, which is very unique for an individual and for identification of the criminal. Therefore, it is revolutionizing evidentiary in DNA forensics and in crime investigation. So, as touch DNA offers numerous advantages, like it requires minimal sample and it requires very less time. It is rapid analysis and quick results are obtained from it. The performance doesn't take much time. It is enhancing the sensitivity and accuracy of the results. But as we all know, every coin has two sides. Along with the advantages, it comes with a lot of limitations. Like there is a possibility of contamination of touch DNA, as our environment has so much microbes in it, so the DNA can be contaminated. It is also dependent on the intensity and time of contact and the duration. For example, if the sample is collected after 2 days, 3 days, the sensitivity of the results is decreasing day by day. The intensity of touch, if there is a single touch, then there is less amount of sample present. There can be contamination due to microbes and environment. Then many other factors are responsible for it. Even there is a possibility of secondary transfer, which is transferred from an individual that the criminal has met before the crime, and it might lead to misinterpretations. So despite of all these challenges, the scientists are trying to improve these limitations, and this touch DNA is actually a revolutionizing evidentiary in DNA forensics and crime investigation. And this touch DNA technique has brought a lot of profound impact and have solved so many cases which was before thought that it is very unsolvable, but those cases are reopened and resolved because of this technique. At last but not the least, I'll just say that DNA analysis is bringing justice close to the victims, a hope for a victim, and taking guilty in account. So touch DNA is the witness which speaks at higher volumes, and it helps to reach the justice within and bring the truth to the light. Thank you for the patience listening and giving me this opportunity to speak. Have a nice day, everyone.